Hello, class. This is your professor, Daryl Fisher. Uh, Chapter 27, Human Development and Heredity. Human Development and Heredity. How do we become human beings? Right. So, in the beginning, start off, human development begins at fertilization. So, remember, the egg and the sperm fertilize. At that point in science, that is a person, has all the DNA that it needs to be able to continue through life. So you have the sperm, you have the egg, fertilization happens, and at that point, life has started as far as science is continued. I know with some people want to argue outside of science when life begins, but as far as science begins, we start at that point because that single celled organism has all the DNA it needs, and now it's going to just start to divide and form into the human beings that we are. Developmental biology studies changes in form and function of the fertilized egg through old age, and then embryology studies the events that occur 38 to 40 weeks of development inside the mother's womb. We have a prenatal period. This is occurs during 38 weeks within the mother's womb. Uh, we usually call that being pregnant. And so that is the prenatal period. Then we have postnatal, which is after birth, postnatal period, which is the majority of our lives. Process of prenatal development, uh, pre-embryonic period for about 2 weeks after fertilization. So sperm, egg come together, form a zygote. It divides by the process of mitosis many times, forms a blastocyst, implants into the endometrium of the uterus, pre-embryonic, and then we have embryonic from week 3 through 8, the embryonic period. The conceptus grows, folds. Remember, it has all the DNA that it needs. It basically is just growing. Folding forms rudimentary organs. Now it's called an embryo. The final stage, the fetal period, from week 9 to week 38, or until birth. Remember, birth can happen a little bit earlier, it can happen a little bit later, but at that point we call the baby a fetus. It grows larger, continues to develop, organ systems begin to function, and eventually will function without assistance from the mother. And then after it is born, we call it a newborn. So pre-embryonic, first 2 weeks, embryonic, weeks 3 through 8, fetal, weeks 9 through 38, and then newborn. The baby's here, baby. That is a diagram of what is happening, developmental stages. Postnatal period extends from birth to 1 month of age. We have this postnatal rapid development. If any of you have children, you know that baby goes from, not even really being able to see you to being able to see and make sounds and move and all these kinds of things. Infancy continues through, we call it child as an infant through 2 years of age. Childhood lasts uh, until puberty. At this point, we begin adolescence. That's a very hormonal time of life. And then we have what is called adulthood, from adolescence till what we call death or the end of life, or when all your metabolic reactions stop. All right, so let's discuss this pre-embryonic period, fertilization through implantation. Right, so pre-embryonic fertilization happens. The sperm and secondary oocytes, egg, form a zygote, continues until blastocysts moves into the uterus and implants in the endometrium wall, that nice wall of the uterus where it can start to receive nutrients. Some cells of the blastocyst begin to form an extra embryonic membrane, so to support that embryo. Uh, Meiosis, remember meiosis and mitosis? Remember, meiosis produces the gametes, or sex cells, which are genetically unique haploid cells with one half of the chromosomes. Gametes combine and form diploid. So the sperm and the egg both have half. They combine and form the zygote, which has all the necessary DNA. So both gametes contribute equally to the number of chromosomes. Therefore, the genetic makeup of the new offspring is a combination of mom and dad. Sperm, as we know, must travel a long distance to reach the oocyte. It needs to be small and motile. Sperm need to swim and move. Secondary oocyte is relatively large, must provide cellular machinery necessary for first week of development. You have cilia lining uterine tubes. Gentle peristaltic contraction propel the oocyte toward the uterus because that egg is not motile or mobile in and of itself. Sperm cells get ejaculated in semen when the man and the woman have come together, which is a mixture. That semen is a mixture of sperm and fluid secretions. The ovulated secondary oocyte is viable for about 24 hours when the woman can get what we call pregnant after it is released from the ovary. Pregnancy most frequently occurs when the sperm enter the female reproductive tract during a 3-day window, 2 days before ovulation, 1 day after. But sperm can remain viable for up to 5 days in the female's reproductive tract. So I may not get pregnant right away, but it could happen eventually. Several events must occur before we can have this fertilization between the sperm and the egg. Before we can fertilize that egg, sperm must undergo capacitation, some functional changes that make it fully motile, modifies plasma membrane. Remember, sperm is still a cell, modifies plasma membrane so it can fuse with the plasma membrane of the oocyte. Remember, the plasma membrane is semi-permeable cells, semi-permeable. Another word, can't think of the word. Look at me, teacher can't think of the word. Remember, it's semi-permeable. Plasma membrane forms the outer covering of the cell, in this case, the sperm. It must fuse with the cell of the egg. So they must fuse, and so it has to undergo some functional changes to be able to happen. So sperm undergo this capacitation as they migrate to the egg, moved by this whip-like flagella. You've seen sperm with the long tail at the end. Prostaglandins and semen are thought to stimulate uterine contractions. The male and the female bodies sort of, think about it, the bodies are sort of working together to make this happen. Sperm moves farther into the female tract. Female orgasm is not required, little lo and behold, but it can still happen to where the sperm migrates to where it's supposed to be, by the efforts of the sperm and male orgasm. It can get to the egg. What we have here is acrosomal reaction. So releases enzymes from the head of the sperm. The ovulated secondary oocyte egg is surrounded by a layer of granulosa cells called the corona radiata. Sperm must penetrate this cell layer by a combination of movements. Hyaluronidase enzyme is released from the membrane-bound sac. The head of the sperm catalyzes this reaction and breaks down the protein layer that serves as the barrier between granulosa cells. So think of it as, you know, this, trying to put this in a respectful way, but the sperm has to sort of break down the walls of the egg so that they can combine. So deep to the egg's corona radiata is a zona pellucida, a layer of extracellular matrix secreted by the oocyte, contains sperm binding receptors. So once we break down that outer layer, we have this next layer that has sperm binding receptors. When the sperm cell binds, the sperm binding receptors in the zona pellucida, calcium ion channels present in the sperm's plasma membrane, open, allowing calcium ions to flow into the cell. Remember, calcium is used in different ways in the body to be able to send signals. This increased intracellular calcium ion triggers acrosomal reaction, releases acrosomal enzymes, catalyzes reaction that digests holes in the zona pellucida. Numerous sperm cells are needed to release enough of this enzyme and acrosin to break through the corona radiata and zona pellucida. Right, so we have to break through this outer layer of the egg. I guess the egg is playing hard to get, so to speak. But when the first sperm cell penetrates the zona pellucida and binds to that egg's plasma membrane, it changes the egg, and zona pellucida. Changes in that egg and zona pellucida occur so that no more sperm can enter. Right? So finally, it takes multiple sperm to release hormones to break down the outer barrier. But once one sperm is able to break through and bind, now no more sperm can bind. He's the lucky one. So the sperm entry stimulates a cortical reaction, destroys sperm binding receptors. All right, so that only one sperm with half the chromosomes of the male binds with one egg that have half the chromosomes of the female. And now we have one single-celled organism that has a full chromosomal set, and that will start to divide, and become a human, continue to become a human being. Next, we have the sperm nucleus swells to form male pronucleus, plasma membrane. Sperm bonds the plasma membrane of that egg. The two membranes fuse. Cytoplasmic contents of the sperm, including the nucleus, are released into the egg. They're becoming one. The oocyte, the egg, completes meiosis 2, and resulting ovum swells to form female pronucleus. At the same time, fusion of the plasma membrane signals secondary egg to complete meiosis 2 and become ovum. Released polar bodies expelled, sometimes means visible edge of ovum, and ovum's nucleus also swells, becoming female pronucleus. Right, so they're becoming one. Spindle fibers between male and female, pronuclei. Pronuclear breakdown, allowing chromosomes to mingle. Remember, we have half the chromosomes from the man, half from the female. They need to come together to make a whole. If any part of this process is interrupted or defective, reproduction will not be successful. Infertility, which is a word you probably have many have heard of, is the inability to produce pregnancy after a year of unprotected sexual intercourse. So having sperm introduced into the woman and yet no viable pregnancy after a year of having doing this process is what we call infertility. But normally multiple sperm will come in, an egg will come down, they'll bind, become one, become a baby. All right, so this is the big picture of fertilization. As you can see, one sperm eventually, ideally, will break through, and then their DNAs will combine, and they'll become a single cell organism with a complete set of DNA. Approximately 30 hours after fertilization occurs, the zygote begins to undergo cleavage. Now we have mitosis. Breaking mitosis, remember, is splitting of the cells with an equal number of DNA in each. So a full copy of DNA in each, sorry. So a series of rapid mitotic divisions that produce small, genetically identical blastomeres. So we're just taking one cell that has all the chromosomes and making it two cells. Each cell has all the chromosomes, making it four cells. All four cells have all the sets of chromosomes. So the division occurs too quickly for cells to grow. So cell number increases, but cell size becomes progressively smaller. So just making a bunch of cells. Divisions take less time. By second day, there are four cells. By the end of the third day, there are 16. At this stage, cells start to differentiate. Differentiate means they're starting to become different parts of the body. By the time the blastocyst is ready for implantation into the uterine wall, it has two distinct cell populations. Outer layer, large flattened cells, trophoblasts surround fluid-filled cavity. They participate in forming the placenta, temporary organs, temporary organs that provide nutrients and oxygen, suppress the mother's immune system so that the mom doesn't attack the embryo. Remember, the embryo is a different DNA than the mom, so it's technically foreign. But you also have this inner cluster of rounded cells, embryoblasts, forming the embryo, which will develop into the body. That is the structure there. Occasionally, within the first 8 days after fertilization, cells separate into two groups, develop into two individuals called monozygotic or identical twins. Cells with the same DNA, same number of chromosomes, divide into two separate groups, twins, genetically identical. Their appearance is going to be similar, and oftentimes they share the same placenta. You could do it again and divide and be triplets or quadruplets. You also can have dizygotic twins, which is a more common type of twinning resulting from the ovulation of two secondary eggs at the same time. So a sperm and an egg and another sperm and an egg happen to get fertilized at the same time. They're not twins, even though they have similar DNA. They're not twins necessarily, just two separate eggs that got fertilized at the same time, now forming two different individuals. Implantation occurs approximately 4 to 7 days after fertilization, when the blastocyst begins to attach to the endometrium of the uterus. Remember, the endometrium is plush with everything that the embryo is going to need. The trophoblast invades the stratum functionalis, the layer of endometrium, by secreting digestive enzymes that catalyze reactions to degrade endometrial lining. Trophoblast divides into two layers: the inner cytotrophoblast, the outer succinktotrophoblast. However you want to say that. Practice that. But the inner cells remain intact. The outer cells, plasma membrane disappear. Cytoplasmic contents merge to form single large multinucleated cell. It begins to secrete enzymes to digest uterine cells almost immediately after formation, so it can bind to that uterine wall. That outside layer is also responsible for secreting human chorionic gonadotropin, HCG, which will stimulate the corpus luteum and ovary to secrete estrogen and progesterone, which promotes the placenta development. Progesterone suppresses menstruation. Remember, we don't want to menstruate anymore while we're developing this child. So we maintain the endometrium of the uterus. Don't want to get rid of it. Don't want to menstruate. Maintain it. Keep that so that the baby has a nice place to grow. The HCG is produced only during pregnancy, making it the basis for urine pregnancy tests. If you have HCG being secreted by your trophoblast, that makes you pregnant. Body would not want to keep the endometrium of the uterus if it didn't need to. So something that is released to keep the endometrium of the uterus for the purposes of the growth of an embryo means that you're pregnant. So you urinate, they test for the HCG, the test comes back positive for HCG, means you are very, very likely to be pregnant. The inner cell mass is the developing offspring, differentiates into two layers, epiblast, hypoblast. The bilaminar embryonic disc will eventually become three primary germ cells, differentiate to produce all tissues in the body, ectoderm, mesoderm, endoderm. Also have the future of organ development. If for some reason the conceptus is not viable, the baby at this stage is not viable, some chromosomal abnormality, for instance, it could die. Uh, and this process is called spontaneous abortion or miscarriage. Some of you, I'm sure you've all heard of miscarriage or may unfortunately have experienced it, but as you can see, there's so many moving parts and pieces to the development of a fetus. And if something doesn't happen correctly, or if things, just don't work out, pregnancy will end. Body can end the pregnancy. There are rare occasions where the blastocyst implants somewhere besides the uterus. It's called an ectopic pregnancy. In that case, the fetus usually will not survive as well. And unfortunately, that can cause problem killing the mother. You just think of the baby developing in the tube, for instance, where it's not supposed to develop and causing internal bleeding as it grows and breaks open that fallopian tube or somewhere else where it's not supposed to be developing. Cause internal bleeding. That internal bleeding could cause mother and child to perish, unfortunately. So there are many potential issues that could happen over the course of fertilization to childbirth. This is a diagram of implantation. By the development of extra embryonic membranes, right? So we have this bilaminar embryonic disc and trophoblast together produce the extra embryonic membranes. They appear during the second week of development. We have a yolk sac, amnion, lantois, chorion. Forgive me for not pronouncing mispronouncing some of these words. I don't use them very often. Um, these membranes protect the embryo. Now think of this, think of the embryo. What is the purpose? We are reproducing life. Male, female come together, sperm, egg come together, fertilization happens, chromosomes bind together, form a full set of chromosomes. Mitosis starts to happen, embeds in the wall of the uterus. We need to protect this embryo as it grows. The goal is to reproduce life. So we have to protect this embryo. This embryo initially cannot function on its own, can't do everything by itself, um, can't eat, can't breathe. And so all of these things are going to have to happen till the embryo gets to the point to where it can do these things on its own. Even after childbirth, they have to get the child breathing, have to help the child with eating. This is a process that's going to happen, take some time to get everything to the point to where this becomes an independent person. Some of you might argue they don't become independent until they're like 20 something, but yeah. So in the embryo, we need to protect the embryo, assist the embryo with vital functions, including nutrition. The embryo needs nutrients, gas exchange. Remember, anything that's living needs oxygen, needs removal of carbon dioxide, uh, other waste, uh, just like any other cell, tissue, or organs in your body. They produce waste. That waste needs to be removed. The yolk sac arises from the hypoblast cells, first extra embryonic membrane to develop. Cells from the yolk sac form part of the digestive tract, which are the source of first blood cells and vessels. Got to have nutrients. Also produces germ cells, precursors. Gametes will eventually migrate in developing ovaries and testes. The amnion, transparent membrane that develops from the epiblast, will eventually enclose the embryo in a fluid-filled amniotic cavity. I'm sure you all heard of the amniotic sac, but this fluid-filled amniotic cavity, penetrated only by the umbilical cord. It'll connect the embryo, and or you want to call it the fetus, to the mother's blood supply. It secretes this sac that surrounds the embryo, secretes amniotic fluid. Fluid is formed by filtering the mother's blood. So think about that connection between the mom and the child. We filter the mom's blood and make this amniotic fluid. Embryo's own urine contributes to some of the amniotic fluid volume once its kidneys become functional toward the end of the 8th or 9th week of development. The amniotic fluid protects the embryo from trauma, keeps it from drying out, maintains constant temperature. We want to keep that baby nice and warm, allows freedom of movement, important for muscle development, prevents body parts from adhering to each other. Amniotic fluid, filtered blood, and eventually urine. Interesting. Allantois, small outpocketing of the caudal wall of yolk sac develops during the 3rd week. We have early blood vessel formation. These become umbilical vessels that will connect baby to the placenta. Tissue around this structure becomes connecting stalk, forms remainder of umbilical cord. We have the chorion, outermost extra embryonic membrane develops from the cytotrophoblast and the other blasts and closes all other membranes in embryo. By late week 4 development, the chorionic villi are blended with the stratum functionalis layer and begin to form principal embryonic part of placenta. Here's where the placenta gets made. Right, so this is how the structure starts to look, starts to form. Amniotic cavity, embryo, chorion, yolk sac, future umbilical cord connecting mom to child. See that there. Yolk sac, future umbilical cord. See that placenta. All right. Week 3 through 8. The embryonic period begins at the 3rd week of development and continues through the 8th week, where we call it an embryo. Generally, we use the word embryo the entire time, but or fetus. We use those kind of interchangeably in the real world, but technically, the embryonic period scientifically is week 3 through week 8. The blastocyst is fully implanted in the uterine wall. The inner cell mass has differentiated into the bilaminar embryonic discs, and extra embryonic membranes have developed. During this 5-week stage, conceptus is referred to as embryo. Embryonic period starts with a process called gastrulation. We rearrange migration of cells of the bilaminar embryonic disc to form trilaminar embryonic disc. Trilaminar 3 layers, These 3 germ layers will become all the major organ systems in the process of organogenesis by the end of week 2. So notice what's happening. Week 3, this embryo, week 3 through week 8, and we will have developed all the major organ systems. 8 weeks. Amazing. Placenta also forms during this period, provides nutrition and oxygen to the embryo, removes waste. But back to the organogenesis. By the end of week 8, by the end of this embryonic period, we'll have formed all the major organ systems. Think about that. All right, so gastrulation, week 3. You don't need to know every single detail to these steps, but just know them sort of briefly. So week 3, primitive streak thin groove on dorsal surface of epiblast, elongates along the cephalic, caudal, head tail line of the embryo. We establish the head, the tail, the right and left sides, dorsal and ventral surfaces during gastrulation. First cells migrate, come inner germ layer, endoderm. We have the mesoderm, you're in the middle, and then the ectoderm, outer layer. By end of week 3, all 3 primary germ layers will be formed of the embryo. So 3 layers. That's the diagram for that. Amazing to see the formation. Now, I want you to see now is organogenesis. 3 primary germ layers differentiate into organs and organ systems. right? So those three germ layers, endo, meso, ecto, will differentiate into the organs and the organ systems. So organogenesis begins. Primary germ layers have undergone cephalocaudal and transverse folding. Now they're differentiating, they're forming the different parts of the body. So when the embryonic period ends at week 8, embryo will be approximately 25 milliliters, about 1 inch long. Will have recognizable organ systems, although not all organs will function. So the ectoderm, the outer layer, forms the epidermis of the skin and the nervous system, majority of the nervous system, and your sensory organs. Okay. Know that. Okay. So we have the anterior end of the neural tube will become the brain, remainder will become the spinal cord by end of the 4th week. Anterior neural tube develops into 3 enlarged areas: primary brain vesicles, forebrain, midbrain, hindbrain. We also have the neural crest cells, cranial, spinal, sympathetic ganglia. Remember that from the nervous system. Cells of the skin, cells of the adrenal medulla, connective tissue cells, mesodermal cells, vertebrae to the ribs, dermis, layers of the skin, and skeletal muscles. These are all from the mesodermal cells. This is not no longer the ectoderm. This is the mesodermal cells. Um, so we have the vertebrae, dermis layer of the skin. Remember, the dermis layer of the skin is beneath the epidermis, skeletal muscles. We also have the intermediate mesoderm forms the gonads, your sex cells, and your kidneys for your urinary system. Lateral plate of the mesoderm forms spleen, adrenal cortex, cardiovascular system, right? So ectoderm, outer layer of skin, nervous system, mesoderm, vertebrae, uh, connective tissue layer of skin, skeletal muscles, kidneys, spleen, adrenal cortex, cardiovascular system, connective tissue of limbs. And then endoderm, your inner layer, forms it's going to give rise to your middle ears, auditory tubes, your epithelial layer of your digestive, respiratory, urinary, and reproductive systems. A lot of your glands form from your endoderm, right? So we have these 3 layers which are going to divide into these 3 different, into well, not 3 different, into all these different organ systems. So by the end of the embryonic period, organogenesis has occurred. 4 weeks, embryo has 3 primary brain vesicles. By 8th week, embryo looks essentially human. And during this period, weeks 3 to 8, embryo is especially sensitive to teratogens, birth defects, even death. So once again, this is a very specific process. Anything goes wrong, it could be very problematic. That's what's happening. Week 4 to week 8. By that time we get to week 8, we can take pictures and see the baby.